So good night, everyone. So we are back with our symposium, with our international webinar of beef quality. First of all, I would like just to clarify what happened yesterday. We really don't know what happened. We had some technical issues. So YouTube just stopped to streaming our live. So if it happened today, what we are going to do, we are going just come back to the YouTube channel. We'll have a new link, so you could click there, and we are going just to continue with the presentation or with the questions with our invite. Okay. So again, it's a really pleasure to have all of you with us. And I'm going just to remember you that this webinar is being organized by the Carcass and Meat Quality Evaluation Research Group, known as the GPAC, under supervision of Dr. Ana Maria Bride from the Universidade Estadual de Londrina. The GPAC was created in 2006 by Dr. Ana Maria Bride and is currently composed of 15 undergraduate students and 7 graduate students in animal science and veterinary medicine from Universidade Estadual de Londrina. The GPAC, they conduct research on pork and beef quality, and as well as on fish meat quality. The group works in a close collaboration with the Brazilian meat industry in the state of Paraná. And just to let you know a little bit more about our university, the Universidade Estadual de Londrina is located in south of Brazil in the state of Paraná. And Paraná is one of the largest meat producers in the country. More precisely, they produce more pork and poultry meat. So today we are already on time. We are going on with the second speaker of this symposium. So today we have the pleasure to receive Dr. Minh Du. He's gonna give a presentation on how can epigenetic and fetal development help improving the marbling fat deposition. So don't forget that following the presentation, we are going to be live with our speaker for questions. So see you soon and enjoy the presentation and prepare your questions.
Okay, thanks for invitation for me to attend this webinar. So today I'm going to talk about epigenetics and fetal development, which can help to improve the meat quality. So we know that different cattle breeds have different growth potential and meat quality, such as the Angus cattle are usually produce better meat than most other cattle breeds. And another example, the Japanese Wagyu cattle are especially selected for their extremely high marbling fat. So they produce better quality meat. So these examples show that cattle genetics affect beef quality. And we know that domestic animals are extensively selected for their lean growth. But not only genetics are important. Epigenetics, which are referring to changes in genome, which does not associate with DNA changes. So those epigenetic changes are inheritable, and they usually generate long-term impacts on the cell differentiation and tissue development. And so the epigenetics are affected by maternal nutrition. Therefore, not only cattle genetics are important, proper nutritional management are equally important because they affect epigenetic changes, which can affect the growth performance and the meat quality of the resulting animal. And we know that animals are more susceptible to change during active development. So for example, here, if we compared the mature cows and her offspring calves, so we know that offspring calves would be more susceptible to stress and nutrient deficiency because this little calf is actively growing, right? But even more vulnerable is that before those calf was born and they are still in the maternal womb, they are even more susceptible to nutrient changes because they are developing rapidly and they are changing rapidly, and any change in nutrients to the muscles could impact the fetal development and their epigenome, which can generate long-term consequences on the offspring of the birth, including their growth performance and beef quality. And for us, the most important thing is skeletal muscle, because this is what we raising meat animals for. We want to get the skeletal muscle, which provide meat. And the skeletal muscle development, in particularly, occurs in the fetal stage. Actually, for beef cattle, the muscle fiber formation occurs before the end of the second trimester, so about the day 210 of gestation. So this is quite early. And after that day, the end of the second trimester, all muscle fiber formation are done. So further growth in the fetal muscle is due to increase in the individual muscle fiber size and length. Okay? So that indicates the pregnancy stage, especially the middle gestation stage, is critically important for the fetal muscle development that could impact long-term growth performance of the resulting animals. Furthermore, we also know that adipose tissue development occurs in the middle to late gestation and continues after birth. And the maternal nutrition, therefore, has dramatically impacts on the fetal and the neonatal growth, especially skeletal muscle and the adipose tissue development, because they are low priority in nutrient partitioning. So this means that the muscle and fat, when there is nutrient insufficient or nutrient deficiency, the animals, so growing fetus, would first use those nutrients to meet the demands of a growing brain, heart, internal organs, under the expense of adipose tissue and the muscle. So that makes muscle and the adipose tissue are especially vulnerable to nutrient deficiency. Okay, so that makes maternal nutrition are especially important in order to improving the growth performance of offspring animals and the meat quality. So this is roughly shows the beef cattle, their development. So we use the conception as the start of life. So to the harvest of the and the end of beef cattle. So we can know that there's middle gestation and both and about 205 days and 250 days. So this is covering the whole period of fetal development. Then about the weaning, days of the weaning, and then of the harvest. So we can see that for the muscle development, the muscle fiber hypertrophy, hyperplasia hypoplasia means increase in muscle fiber number, which are mainly occurs during the first 2 trimesters, so about day 210 days of gestation. Okay, this is a major period of muscle fiber formation. Then afterwards, muscle growth is mainly because muscle fiber increase in diameter and the length. We call muscle hypertrophy. So this is the transition from hypoplasia, increase in number, to the increase in the muscle fiber sizes in the later development. So this is a muscle. And when look at the fat, here I separate intramuscular fat, which are marbling fat, from other fats. Other fats is referring to the visceral fat, the fat inside abdominal cavity, and then subcutaneous fat, and the thin fat or intermuscular fat. All these fats are not commercially important because they did not contribute to the eating quality of the meat. So I group all these other fat together, and compared with the other fat development, the intramuscular fat development is slightly later, occurs slightly later. And then their increase in number or hypoplasia, as shown here, the yellow stage, continues to about 250 days postnatal, okay, after birth. And then they largely switch to the hypoplasia, increase in size. So if we want to increase in the marbling fat of the beef cattle, it is better we provide better nutrients during the stage where intramuscular fat is actively developing form, because those newly formed adipocytes, they can provide areas where future lipid can be deposited, such as the lipid deposition during the fattening stage, during the later stage. So this, in the end, would result in high marbling beef. Okay? So as I said, this pregnancy stage is really important regarding the fetal cattle development, which could induce long-term changes in growth performance of offspring cattle. Because beef cattle pregnancy is relatively long, this provides opportunities for stage-specific nutrient manipulation to enhance offspring growth performance. This is especially the case for the muscle and adipose tissue. However, nutrient deficiency frequently occurs in cows due to the seasonal reproduction of cows, and which usually coincides with early spring. And this stage, the forage availability usually low, so the cows could frequently experience nutrient deficiency, which could impact muscle growth and the overall meat quality. So as shown here, this diagram again shows the conception to both, so covering the whole fetal cattle development. So this shows during the early stage, 1 to 3 months, there is we referring to as embryonic stage, where primary myogenesis occurs. Then there's a major formation of muscle fibers during the secondary myogenesis, which occurs between 3 to 7 months of gestation. Then the muscle fibers are mainly characterized by increase in size. And the nutrient deficiency during different stages have different impacts on the growth performance of offspring cattle. So nutrient deficiency during the first and the second trimester, which corresponding to the myogenesis muscle fiber formation, which can reduce muscle fiber numbers and the muscle mass in offspring. But nutrient deficiency later in fetal development only decrease birth weight, and the muscle growth likely be recoverable because the number of muscle fibers is not affected. And the nutrient deficiency, however, during the late fetal stage, where adipogenesis occurs, the nutrient deficiency during this stage could reduce the modeling fat cell development, which could impact the modeling of the offspring. This also including the neonatal stage and the before weaning stage, because the adipose tissue, especially intramuscular adipocyte formation, continues after birth until pre-weaning stage. So to test a number of, about 10 years ago, we did a study in the cattle where we use native range and improved pasture. And the native range is in the area of Wyoming. So this study was done in the Montana and Wyoming area, where in the United States, this is the area where it's very dry, so there are limited forage available. So during the native range, that means the forage availability during the middle gestation became quite limited. And then we have improved the pasture, where those pastures are seeded and we irrigated, so they provide much better grass quality. So this results in two different nutritional levels. One is lower crude protein levels, okay? And one is high crude protein levels. So this meets the nutrient requirements of the cattle, and the native range is nutrient deficient for fetal development. So this was applied in the middle gestation for about 2 months, between 120 to 180 of gestation. And so this is corresponding to the middle gestation of the animals. And this actually resulted in significant impacts on the fetal development. Let's look at the weaning weights are significantly different. By providing better nutrition during the middle gestation, we got weaning weight significantly higher. Final body weight of the offspring cattle was elevated, and daily gain was improved, and the live weight and the harvest was increased too. And importantly, after harvest, the hot carcass weight was higher. That was quite significant. And furthermore, we analyzed the tenderness of the meat. The tenderness was improved as indicated by lower shear force that indicate the tender meat, and the collagen content was low, so this correlate with the improved tenderness, and the ether extract, this reflect intramuscular lipid content was elevated in improved cattle of improved pasture. So all of these in combination, we found maternal nutrition during the middle gestation could impact the long-term growth performance by providing better nutrition during this stage. So cattle growth efficiency can be improved, meat quality can be improved. So this shows that maternal nutrition affects the tenderness of the offspring cattle, so showing the production and the meat quality problems have a fetal origin. So in summary, the maternal nutrition could impact fetal development, which has long-term effects on the growth performance of offspring. And by improving pasture condition or nutritional condition of the beef cattle, we can improve the quality of the meat of offspring cattle, while poor nutrition can reduce growth performance. So this raises a question: if we supplement cows with proteins, what's its impact on muscle growth? Because muscle development requires large quantity of the proteins. So where the supplement of the nutrients, especially proteins, can enhance fetal muscle development. To test this, we did another experiment where cattle were fed 100% meet nutrient requirements or meet 70% of nutrient requirements, or 70% nutrient requirements plus protein supplement to meet protein requirements of pregnant cows. Okay, this was applied from the 4 to 5 days of gestation to 185 days of gestation, covering the whole period of secondary myogenesis, or the major stage of fetal muscle development. And we want to show whether this affects the growth performance of calves. And the steers, resulting steers were harvested and about 1.5 years of age. So we can see that the muscle weight was different. Nutrient deficiency did not quite affect the muscle weight, but by providing better nutrients, we found that the ST muscle weight was significantly improved, and the ST muscle weight as a percentage of hot carcass weight was significantly increased, but the fatness of cows was reduced due to nutrient supplement. This data clearly shows that by providing better proteins during the fetal development, improve the growth performance of the offspring cattle. So this shows that fetal programming through manipulating maternal nutrition has a major effect on the growth efficiency of beef cattle, and also beef quality. This including effect on the lean fat ratio, feed efficiency, and the marbling of the offspring beef, as well as the tenderness of the beef. Okay. So as I summarized here, so we expected the nutrient deficiency during the pregnancy and the different stage have different impacts. So stage corresponding to the muscle fiber formation could affect muscle growth, and those stages affecting the late gestation and the neonatal stage could affect marbling fat development. Okay, so let's focus more on the marbling fat. So marbling is critically important for the eating quality of beef, and the marbling fat development is associated with two factors. One is hyperplasia, referring to increase in number, and hypertrophy referring to increase in size. Okay, as you hear, both increase in the size, both the increase in the number could result in larger muscle mass, so more modeling fat. So for us, the formation of intramuscular adipocytes is especially important because when those intramuscular adipocytes they are formed, they can provide sites for later lipid accumulation, which form intramuscular fat or marbling fat. So as shown here, these are intramuscular, these are the early progenitor cells. So the progenitor cells need first convert into pre-adipocytes, usually occurs along both for the intramuscular fat, and then they become fat cells. Those are immature fat cells. They do not have much lipid yet. Then they need accumulated lipids intracellularly. This results in their increase in size. So the slow hypertrophy then result in development of marbling fat. So there are two factors here. We either increase in the formation of the number of intramuscular fat cells, and we also can increase the size of existing intramuscular fat. So this we call hypertrophy. And this another line shows other depots. As I said, we separate them because they are not commercially important regarding eat quality. Okay, so if we summarize this, we have several points where we can regulate the intramuscular fat deposition or marbling fat development. We can increase the density of the progenitor cells and enhance pre-adipocyte formation and enhance their conversion into adipocytes, and those adipocytes then can accumulate lipids through adipocyte hypertrophy. Okay, so there are 4 critical control points to enhance marbling fat deposition. And in our recent several years, we studied the vitamin A impact on all these 4 different stages. So vitamin A, we think that they have a dual effects on marbling fat development. In the early developmental stage, marbling vitamin A and its bioactive metabolite retinoic acid, they can enhance progenitor cell proliferation. These proliferate to form more progenitor cells. And then these more progenitor cells, they can undergo conversion into pre-adipocytes and then later develop into mature intramuscular fat. So we think during the early development, vitamin A can enhance marbling fat, marbling adipocyte formation. But in the later stage, we think vitamin A actually can prevent lipid accumulation because vitamin A is known to enhance lipid oxidation, which can reduce adipocyte hypertrophy and result in the reduction of intramuscular fat. For this reason, vitamin A withdrawal during the fattening stage is used to enhance marbling fat deposition. So our study want to focus on the early stage of development, where vitamin A can enhance progenitor cell proliferation and their conversion into adipocytes. So to test, and we applied vitamin A during the early developmental stage. And we think this study is important. And also for the Brazil, because in the Brazil, there are large areas are lack of quite a draw in the reproduction stage of cows, will limit the fresh forage availability. And we know carotene is a major source of vitamin A for beef cattle. So lack of green forage with a limited carotene intake, actually severely limiting beta carotene intake, which could result in vitamin A deficiency. So to study vitamin A impacts on the calf development and the offspring growth performance is applicable to the beef production in Brazil. So in this study, we used injection of vitamin A's. We inject the neonatal calves, and then under 1 month old, we inject twice. And we using the injection, so first injection, second injection, because we want to avoid the impact of vitamin A on the cows, which could complicate the interpretation of results. Okay, so after the first and the second injection, we did biopsy and 30 days and 60 days, which we want to evaluate biological changes in the muscle and the marbling fat development. Then those offspring cattle will win and 210 days, and then go through backgrounding diets and the finishing diets and harvest and 429 days. Right? So we can see that by injecting different doses of vitamin A, it impacts the intramuscular fat deposition, okay, which are increased by early administration of vitamin A. And the marbling score was significantly increased too. We also found unexpectedly vitamin A can enhance the daily gain of the calves. The average daily gains was significantly improved due to vitamin A, which resulted in a net gain of calf weight and winning about 20 kilograms. That is dramatic. This 20 kilogram increase in weight is very significant. And this appears to maintain after and finishing stage the weight gain. So it's quite significant. So this study shows that vitamin A administration during early development stage, in neonatal stage, could enhance adipocyte formation, which could provide sites for the marbling fat accumulation, resulting in higher marbling of offspring cattle. In addition, so calf growth was enhanced by vitamin A administration, which was not expected, but this suggests that vitamin A likely enhanced muscle growth, because we found the frame of the animals measured by hip bone height was not altered by vitamin A administration. So this suggests that muscle growth likely enhanced. To show the muscle change, so we analyzed the muscle area in addition to the body weight change. So this slide shows body weight change. We can see the vitamin A increase the body weight gain. Okay, body weight gain. And this we only use the low dose of vitamin A injection for further analysis. And if we compared the biopsy samples of the control and the low dose of vitamin A injection, we found those markers of myogenesis, muscle formation, are all significantly increased, actually dramatically increased in the vitamin A treated group, and those modeling, those muscle growth markers, as shown here, are correlate with fetal formation, early formation of muscle development, and the later formation of satellite cells and their fusion with existing muscle fibers, and overall they contribute to the muscle growth. And all these are dramatically increased due to vitamin A treatment, and this is confirmed by Western blotting analysis. Because PAX7 is a marker of satellite cells, and the satellite cells are critically important for the postnatal muscle growth. So we analyzed satellite cell density, which was dramatically enhanced due to vitamin A treatment, which you might not see very clearly, but this is the brown, the red stain, and I expanded the two stains as shown here. Those are the satellite cells. Okay, so we quantified those stained satellite cells as shown here. They are increased by vitamin A treatment. So enhanced vitamin A induced PAX7 cell density or satellite cell density, likely explain the enhanced muscle growth in vitamin A treated cattle. To further show, we separate satellite cells and then we differentiate them into myogenic cells in vitro. We found that the myogenesis was enhanced by in the vitamin A group, and the more muscle fibers or myotubes was formed in those cells separate from vitamin A group. So this is consistent with enhanced myogenesis in the vitamin A treated calves. Okay? As a result, we found that the ALT muscles and the harvest was have increased muscle fiber diameter, and although the distributions was shifting to the larger size for the vitamin A treated muscle, and the river eye areas and the harvest was larger. Okay? So all of this confirms that vitamin A treatment during the early developmental stage not only enhance marbling fat development, but also can enhance satellite cell development and muscle development. Okay? So this is a very exciting result. So in the end, I want to emphasize the importance of nutritional management, especially during the early developmental stage, to the cows can improve the offspring growth performance and meat quality. So this is a stage commonly ignored or neglected previously, but our studies and studied from many other labs around the world showing the importance of this stage in improving growth performance and the meat quality of offspring cattle. Okay? So again, the long-term gestation of beef cattle allows stage-specific or precision nutritional management. Genetics are critically important in determining growth performance of cattle. And cattle nutrition, including vitamin A during the fetal stage and lactation stage, early lactation stage could be critically important, which can impact the growth performance and the meat quality of the resulting cows. Okay? So we can manipulate the nutrition during early development, which can induce epigenetic changes and affect the offspring growth efficiency and the quality in order to maximize their genetic potential. Okay, so this study was mainly conducted by my previous graduate student. Right now, he is a faculty member in China Agriculture University, Dr. Bu Wang, and also assisted the studies of the beef cattle studies. Nutrient deficiency and the protein supplement was accomplished by Dr. Keith Underwood, who is an associate professor in South Carolina State University in USA. Okay, there are other members in the lab. Okay, so our studies or our labs are mainly supported by USDA and also NIH grants. So with this, I will stop my presentation. Oh, by the way, this is a picture of the University of Washington State. And we have a quite beautiful campus. So if you can visit our campus, and always welcome to visit my laboratory. So thank you, and we will, if you have any questions, we will discuss later. And bye.
So hello everyone, we are back. Just remember that in a few minutes we are going to be live with Dr. Du. But before that, let me introduce myself again. My name is Luyen. I am graduated in animal science from Universidade Estadual de Londrina, and I have a PhD in animal welfare and meat quality. So I would also like to thank all the backstage from the GPAC group for this awesome work that they are doing. They are working so hard. So thank you very much, group. And just to remember you, if something happened during the question session, if the streaming just fall off, please come back to the channel and a new link will be available so you could just continue with us. So now I would like to introduce Dr. Mindu. So is he? Hi, Dr. Mindu.
Hi.
How are you doing?
You're good.
So first of all, in behalf of everyone here, I would like to thank you for the really great presentation that you prepared for us. It was really interesting, and I'm pretty sure that we are going to have lots of questions on that.
Yes, thank you.
So before I start with the questions, I would like to talk a little bit about Dr. Du. So Dr. Du, he has an honors bachelor's degree from Zhejiang University in China. He has a master's degree from the Faculty of Food Science and Nutritional Engineering from the China Agriculture University. He has a PhD in biochemistry from the Faculty of Biological Science, China Agriculture University. He has a PhD with distinction from Iowa State University. He is currently associate professor at the Faculty of Molecular Science and the Faculty of Food Science in the Washington State University. He is also the professor and the founded chair in the growth biology in the Department of Animal Science in the Washington State University. And Dr. Du received a Research Excellence Award from the Washington State University of Agriculture, Humanities, and Natural Resources. He also received an award in animal growth and development from the American Society of Animal Science. So his research field is more for epigenetic regulation of stem cell differentiation, myogenesis, and adipogenesis. So it's really a pleasure to have you with us today, Dr. Du. So I'm going to open the chat. So if you have any questions, you can start right now. So yes, we have a question. Does hyperplasia and hypertrophy in the pre and postnatal phase have any variation between both torus and both indicus?
So I'm guessing you are asking about the hyperplasia and the hypertrophy of fat cells, so adipocytes. I would say that I'm not sure because as I will say, is no direct study comparing both torus and both indicus about the hyperplasia and the hypertrophy difference. But I would say they should have some difference because we compared the high morbidity breed Japanese Wagyu cattle that's a high morbidity and compared with Angus, and we found there is a significantly higher hyperplasia of adipocytes or fat cells, intramuscular fat cells in Wagyu cattle, so Japanese high morbidity cattle. So I guess yes, but there is no direct study conducted as I'm aware.
Okay, that's good. I hope it answered your question. So do you have some more questions? Okay, so how could be explained the reduction in collagen content in the system with better coal nutrition and change in collagen solubility?
Yes, this is something we are still doing study on. I will say that based on the current study, the adipocytes or the fat cells and the connective tissue cells or fibroblast cells, they share a common group of progenitor cells. So those cells have a capacity to either go adipogenesis or form fat cells or form fibroblasts, which synthesize collagens. So our study shows that better nutrition would drive their differentiation into adipocytes, which form more fat cells inside the muscle. So that's end up have a higher morbidity and correspondingly reducing those cells which synthesize collagens, so reduce the collagen accumulation and the cross-linking. So that what we are thinking that could occur.
Okay, that's good. So we have another here. Thanks for the lecture. So increasing the adipogenic differentiation and reducing fibrogenic differentiation from progenitor cells was what conferred to Wagyu breed excellence in marbling?
Partially, actually not entirely. So we found actually in the Wagyu cattle, overall, they have a higher density of those progenitor cells. So that resulted in higher morbidity, actually, although higher connective tissue in the Wagyu cattle. Both of them are elevated because the progenitor cell density are elevated. However, somehow for unknown reason, which we are still examining, is that the cross-linking in Wagyu actually not increased. We compared with the Angus actually are slightly reduced. I do not know the reason why, but we are still studying. But that's quite interesting thing to further study to illustrate.
Okay, that's good. So, Dr. Du, I know it's hard to say, but what is the responsibility of the animal's genetic background and fetal development to increase IMF marbling? So the intramuscular fibers for marbling.
So this is. So I would say that the marbling certainly are determined by two factors. One is genetic, so that I think in the question called genetic background, and also so fetal programming is referring to the nutrition side, the environmental factors. So genetics certainly have a dominant role in determining the animal's capacity to increase marbling or their growth potential. That's the reason why we selecting animals for better genetics. However, in order for the genes to properly express or achieve the full genetic potential, I think nutrition is essential too, because different nutrients and environmental factors will determine which gene to express and how they are expressed, or to certain degree, what degree they express. So all of this would be affected by nutrition, especially in the early developmental stage when the fetus is highly susceptible to nutrient availability. So I will say that this is the interaction between genetics and the nutrition and other factors. So in order to achieve best growth performance, we need both
Okay.
genetics and nutrition.
Okay, so
Yeah.
that's nice. So we have another question here. Does oral vitamin A administration in pregnant cows or in calves can result in any similar effect?
Yes, I would say so. I aware some groups already studying and publishing related results. And they fed vitamin A during the late pregnancy, and they found that the marbling fat was enhanced. So I say yes, they can achieve similar results. So reason in our study we are using injection, because the injection first is easy to administer, because and both anyway we need vaccinate the cows, so we can do vitamin A injection at the same time as a vaccination, so there's no increased level cost. And of course, for the neonatal calves, supplementing vitamin A would be difficult because they are drinking milk, so not directly intaking feed. But certainly we can administer the maternal cows with the vitamin A. The reason in this study we do not use a maternal supplement of vitamin A is because that could also change maternal milk production, or some other factors could be changed, which of course a portion of vitamin A would be transferred to the calf through milk or through pregnancy, but that would be more like an indirect effect. So that's the reason we did not use it, because that could be complicating the interpretation of the results. Instead, if we're using direct injections, that's more direct. We know this is directly exerts impact on the cow development, calf development. So that's the reason why we use that. But I do believe they can achieve similar effects.
Okay, that's really interesting. So we have
Mhm.
another question here. Good evening, Dr. Du. My question is, a massive tick infection in cows during the first month of pregnancy can cause effect in the development or in the deposit of marbling fat of the progeny.
I I'm not sure that cause. Yeah, I haven't quite checked those reference, but according to my understanding of the muscle and fat development, because the muscle is mainly developed during the middle gestation, and
Okay.
the fat actually are slightly later, close to the late gestation and the neonatal stage. So I would say that I do not expect that could be significant impact. Of course, if this impact the cow body condition scores, and overall nutrient delivery to the fetus during the development could be reduced. That could impact the muscle development. If that persists to the later stages, then could impact on the marbling fat development. But I would say it's a little hard to say. It's depending on the severity of the infection and it's whether it has lasting effects.
Okay, that's good. So we have here, do you have any amino acids with the potential to modulate fetal development or the B vitamins?
For the specific amino acids, I would say that's less important for the beef cows compared with some non-ruminant animals, because beef cows, they can obtain amino acids from the rumen fermentation from bacteria partially. So I would say that for ruminant animals, the overall protein content or supply is or nitrogen related compound supply might be more important than a specific amino acids compared with non-ruminant animals. As for vitamin B, I think there are already available studies show that the vitamin B supplement can affect marbling fat development.
Okay, that's really good. So is there, oh, sorry. So do you have some more questions from the audience? Please feel free to ask your questions. We have to take advantage that Dr. Du is here with us. So we have another one here. Hello, Dr. Du. Great presentation. So based on your studies, would you suggest what are the main epigenetic markers controlling vitamin A effect on early adipogenesis?
Okay, hello, Márcio. So yeah, that's good. I think that's a very good question. So I would say that vitamin A actually is would be converted into retinoic acid in the body. So retinoic acid is the ligand for retinoic acid receptor and retinoic X receptors. And both of these, actually especially retinoic X receptor is required as a partner for the PPR gamma, which are transcription factor which initiate adipogenesis, and some other nuclear receptors. So as a whole, they could affect their commitment to adipogenesis. That's the reason why we think that the early stage vitamin A supplement actually can enhance progenitor cells, their differentiation into adipocytes or pre-adipocytes, so that result in more formation of adipocytes or adipocyte hyperplasia, and that provides site for later marbling fat development, deposition, and enhanced marbling. So I think that epigenetic wise, because the complex like PPR gamma and RXR, retinoid X receptors, the complex can induce local epigenetic change, which facilitates gene expression, especially the genes related to adipocyte development, so that the overall adipogenic commitment and the differentiation can be enhanced by vitamin A. Okay?
It's really interesting. So we have another one here. Congratulations for the great lecture. Is there any indication of how the marbling fat is distributed in the muscle?
Yeah, that's a trick question. So that's actually depending on the breeds. Like high marbling breeds like Wagyu cattle, actually the intramuscular fat can be distributed between or even inside endomysial. That is a connective tissue circulating individual muscle fibers. So could exist for the high marbling cattle, but for the most cattle breeds, I would say it exists in the adipose tissue surrounding the muscle, primary muscle bundles or secondary muscle bundles. So again, that depends. But if the fat is deposited outside the muscle bundles, we call it thin fat. that mostly we refer to intramuscular fats that might not be part of modeling. So it's dependent on the breed of cattle. There is some difference in their distribution.
Okay, that's good. So I do have a question. So I prepared a question for you. Just after that, I'm going to read the comments. So in a culture like Brazil, where cows spend the most part of the gestation eating low quality pasture, especially during the winter season, what could be strategy using the epigenetics to improve the growth performance of the spring? So it would be better the injection of vitamin A in the cows or in the calves?
I would be cautious about the vitamin A injection into cows, especially in the before middle gestation or surrounding middle gestation. So reason is that if we are using injection, a large dose of vitamin A into cows, that could cause might cause some changes in fetal development, especially during that vulnerable stage. Because when you inject into cows in a large dose, that could lead to a sudden increase in the vitamin A level in the maternal circulation. That certainly could translate into a sudden increase in fetal circulation. That could cause some change, acute changes in fetal development.
Okay.
We haven't checked what kind of change could result in, but the earlier development, like some essential organs are developing, that could be more susceptible to change. But in the later stage of pregnancy, we know that that's mainly muscle and adipose tissue developing, then that might be better, maybe lesser impact on the overall development of the fetus. So for the maternal administration, I would suggest using feeding would be better. That is a more gradual supplement, especially for the Brazil, if that's very dry and there is no fresh forage available. Vitamin A supplement really helps to provide sufficient amount of vitamin A, which is essential for the fetal development and the marbling fat development. But not only vitamin A, if the cows is not getting sufficient nutrients, I would say that also need to be supplemented because the cow need to provide sufficient nutrients for the fetal to develop. So I would say both need to be supplemented.
Okay, so that's really good. Thank you. So we have here another question. Thanks for the presentation. There is any relationship between vitamin A treatment and reduction of intake due to increase in leptin?
I do not have any direct information to evidence to provide it, but I guess based on my understanding, because in the cows we haven't do that, but in the adult cattle, I would say if we treat it with vitamin A, it usually would increase the feed intake slightly. So that would be opposite of leptin, which is a satiety hormone which reduces feed intake. So I would say there is a negative correlation between vitamin A treatment. So reason why I think vitamin A could increase the feed intake in the mature cows or mature cattle is because vitamin A, when under relatively high levels, they enhance lipid oxidation. And when there's enhanced lipid oxidation, they consume more energy. So I would expect which result in mature cows to consume more feed. So there is an approach in the feedlot for the cows in feedlot. There is currently in practice to reduce vitamin A. If we're reducing vitamin A in the final stage of fattening stage, then actually there's less lipid oxidation. Less lipid oxidation would translate into a little higher feed efficiency and also would facilitate fat accumulation in existing intramuscular adipocytes, that actually can enhance modeling because due to adipocyte hypertrophy.
Okay, that's good. So I think we don't have any more questions. So while we wait for the audience, if they just came up with a new questions, I would like to show some comments that our audience has for you, just to express their appreciation for the great talk you've done and your availability to be here with us discussing on this subject. So we have here a comment from Ana Maria Bridi. Congratulations, Dr. Du, on the great lecture. We are very grateful for your presence in this event.
Yes, thank you.
So we have another one here. Congratulations, Dr. Du, on the great lecture and GPAC for this event.
Okay.
And thanks for the explanation, Dr. Du. Hope you are doing great. And thanks for the answer. And one more time, thanks for the great opportunity to watch you.
Yeah.
And we have also Dr. Du, thanks for the clear presentation. Coarse or fine marbling may be manipulated by epigenetics factors.
Yes, I think it's possible. I will say that early development is a stage where fine marbling or the intramuscular adipogenesis can be enhanced, which provide the main insights for the marbling fat deposition in that stage, in the fattening stage. So which would result in finer marbling or the better distribution of intramuscular fat.
Okay.
Yeah.
So I would like to know from my backstage, do you have some questions? Okay, so last minute. In while I just thank you, Dr. Du, for your presentation. Thank you for your availability to be here with us. Thank you for sharing with us this really interesting discoveries, this interesting research that you do. Really, really thank you very much for being here with us. So
Yes.
thank you for your participation.
Yeah, thanks for inviting and give the opportunity to give this presentation. Thank you.
So I'm going to let you go. And I just want to remember everybody that on August 20, we are going to have Dr. Jean-François Roquette, and he's going to talk about, let me just read, I forgot the name of the presentation. He's going to talk a little bit about the will still eat meat from livestock in the future. Well, this is a really interesting topic as well. So thank you very much, everybody, and see you on August 20. And Dr. Du, have a good day. And it was really a pleasure to have you here today.
Yeah, thank you. Okay, bye-bye.
Bye.